And right now we will start with Dr. Megan Rolf. So Megan, after a couple of wrong turns, starting off in Kansas where she's native, ended up through Missouri and Oklahoma and finally made her way back home to Kansas. She started about the same time I did here at Kansas State. She's an assistant professor in the animal science department, and her specialty is absolutely genetics and genomics. She teaches our undergraduate students as well. So without further ado, I'd like to introduce Dr. Megan Rolf, and we'll get kicked off here.
All right, well, right now today the microphone is my disruptive technology because I couldn't get it working. But first of all, thank you all very much for having me here today, and I'm excited to talk to you a little bit about CRISPR. And that is in fact not an adjective that we're using to describe lettuce we just brought home from the store versus what we've had laying around for a while, but this is going to be today a discussion of a genome editing technology. So you may have heard it called gene editing. I don't really like that term. I prefer genome because I don't think it's too accurate. But if you hear either of those things, it's basically discussing the same type of technology. So this all relates back to genetic modification. And we know that we can make genetic modifications through things like natural and artificial selection. This is not what we traditionally think of as a GMO, right? But we can change the mean of various populations just by selecting for the things that we find desirable. And that's basically the goal of selection programs. Now, when we start to think about how we might begin to apply a technology like CRISPR, it's going to be a little bit different. So it's going to fit more into these other two categories on the slide here. So when we think about a genetically modified organism, my go-to is always I'm thinking of a transgenic animal, right? So a transgenic animal being one, or a transgenic plant where we've taken a gene from another species and we've put it in whatever species we're interested in. So some examples I have up here would be a mouse that has human growth hormone inserted in it, so the mouse grows to be larger. Or we have green fluorescent proteins from jellyfish that we put in the mouse genome and make them glow green under certain lights, right? So those would be examples of transgenics or transgenesis. So with sort of thinking about applications of CRISPR and genome editing, we can start, and I think the sort of overall thought process has shifted a little bit to incorporate some more thoughts around cisgenesis, which is basically where we're just trying to take a gene or an allele or some type of mutation within the genome within the same species and just move it to a different animal. So we'll talk a lot more about that as we move on. But if we're really talking about wanting to do some of these things, why not just do it naturally? Why even think about using genome editing technologies or something of that nature? Well, first issue is we can only do that if it's cisgenesis, right? We can only select for something or try and move it into the population if it's in the same species. We can't mate the mice and the jellyfish to get the green fluorescent protein. But if we are talking about cisgenesis, that is an option, right? If we've got a polled allele in Angus and we want to put it in Hereford, we can mate them together and get that polled allele in that Holstein population, right? We can get polled Holsteins that way. Or we could mate the polled dairy cattle to our horned dairy cattle and move that polled allele into the population of horned Holsteins, right? That's a doable thing to do without any kind of genome editing technology. Now, the challenge is if we start to thinking about doing that, let's say we just use the polled Holsteins as an example. So they have a lower level of productivity as a general rule. And the last numbers I was able to look up were that it was about $252 per lactation cycle, okay? Lower for those polled animals. Okay, so let's say we mate them together. We have a sacrifice in production that results from doing that. But then we can go and start the process of back crossing. So then once we have that cross animal that's going to be polled, then we start mating it back to the horned animals, right? And then choosing to select the polled animals from that. And we can eventually, in theory, end up with just the polled allele and then everything else being those original horned Holsteins, right? Now, the problem with that process is that it takes an exceptionally long time to actually achieve this goal. And so the last paper I saw on this looked at about 20 years of breeding efforts to get about 50% polled animals using this type of approach. So this is a challenge where conventional breeding techniques are a little bit slower than using this particular technology. And the other downside is that we might lose some of the favorable alleles that we had in those original horned Holstein populations in this process. So that would be another negative. So what can we do with genetic modification that might solve some of these challenges? So with older types of genetic engineering technologies, we could insert either a transgenic gene, which is probably what most people think about. It's what I usually think about. We could do the same thing with the polled allele. We could do insert the polled allele into those horned Holsteins with this same type of approach. Let's say we just want to add something to this one specific area of chromosome 16, right? The challenge with that is with the older types of technologies, whatever we wanted to put in there, we just sort of had to put it in there randomly and hope that it ended up in a spot that it actually worked. Because if we want to express an allele, we need to make sure that it gets inserted into the genome in a place where it can actually be expressed by the machinery in the cell. And so if we're just randomly sort of pasting things into the genome, as you can imagine, what if we paste it in the middle of another gene, right? We might actually cause the individual to be non-viable if we do something like that. So the bottom line is that with these types of approaches, if you're trying to use this, you may have to take and try this thousands of times, hundreds or thousands of different cell lines to try and get this stuff to work correctly. And the other challenge with it is with these older technologies, you actually have traces of recombinant DNA that could be left in the genome of those transgenic or could be cisgenic, I guess, animals. Okay? So from a standpoint, it's hard to say this is exactly like that if we've got pieces of recombinant DNA floating around in there. If we were to sequence those, they wouldn't look the same. Now, genome editing solves a lot of those particular issues, and that's why it's been so revolutionary and people are talking about it and so very excited about this type of technology. And it works basically like this. So you've got it's called CRISPR-Cas9. So you've got a Cas9 protein here, and then you have these guide RNAs. And basically what that does is the guide RNA says, go here, okay? It's basically the targeting mechanism to say, we want to make the cut or the edit, whatever we want to change in the genome, it's right here. And so using that RNA sequence, we can direct it to that particular location in the genome. And then the Cas9 protein basically works as like little pairs of scissors to cut the DNA in the place that we want it to cut, where the RNA told it to go. And so this was originally came from bacteria. So that's how bacteria can defend themselves from viruses. So we basically co-opted that same mechanism, but now we can use it to do something that we find desirable. So the overall idea of that Cas9 is, if we think back to that picture of the genome with all the chromosomes on it, what that Cas9 does is it goes to the place where the RNA said to go, and it basically chops that chromosome in half. Okay? As you can imagine, cells don't really like it when you go in and take a big cleaver to the DNA in it and chop it in half, right? So that activates all of the DNA repair mechanisms within the cell. And we can co-opt those to do things that we'd find desirable as well. And that's how this whole process works. So we can either chop it in half and maybe let it repair itself. We might do that if we wanted to knock out a gene or get rid of a gene. If we want to insert a specific allele or mutation that we find desirable, we're probably going to be doing what we see here on the right. So you can see it can be a little piece of DNA, or it could be a pretty big piece of DNA, maybe even an entire gene. But we can sort of co-opt that mechanism to repair the cell and to make changes in the DNA that we actually find desirable. And so the cool thing about this technology is that we don't have those traces of recombinant DNA like we had before. So if we're using that sort of cisgenics approach where we're taking something like the polled allele from Angus and putting it in Holsteins, horned Holsteins, if I were to sequence that Holstein animal, it should look exactly like a Holstein, except in that one little place where we put in the polled allele. And that's going to be coming from another breed within the same species, right? So from a consumer acceptance standpoint, you can start to see where that may be a little less objectionable than something like a transgenic organism. And I think because of this, it's sort of been dubbed as precision breeding. So if you hear things about precision breeding, be thinking genome editing. So it has been utilized to do a variety of pretty interesting things within livestock. Probably the most famous one is creating those polled Holstein animals. So this is Spotty Guy and Burry over here on the right. And those, to my knowledge, were the first two polled Holstein bulls created from horned Holstein animals using genome editing technology, using CRISPR. Or it might have been talons in this case, but same sort of idea. They've also taken Nelore cattle and made a myostatin mutation to make them more muscular. They've also done a lot of things in pigs. So using a gene from wild boar, I believe, to make them more resistant to viral diseases, or trying to make the pig more environmentally friendly and excrete less phosphorus in the manure, for example. So these things have kind of been done as a proof of concept, right? We're not out there, these animals aren't out in the industry, we're not eating these things at this point in time. But these are kind of interesting proof of concepts where they've taken natural variation that exists within that species or within closely related species and just sort of moved it into an animal that had other sorts of desirable characteristics. So I've heard this, sort of the use of this technology in the beef industry referred to as sort of like the cherry on the top of a sundae. And I actually think that's a pretty good analogy because we have lots of really effective genetic selection tools already, right? We have our EPDs, which might be like the ice cream in the sundae. We have the ability to use genomic technologies like SNP genotyping to enhance the accuracy of those EPDs. So that might be like the whipped cream. And then we might be able to use something like genome editing, which would be kind of like the cherry on top, where we can go in and change specific things in these animals that we might want to change. Now, the caveat of this is that in this process, we might be able to accelerate conventional breeding methods and basically increase genetic progress. But the things that we're going to be able to target with this type of technology will probably be limited at least for a while. So things that are sort of these large, very obvious Mendelian type traits where we have one mutation that controls the phenotype. So black versus red coat color or horned versus polled, things like that. The other targets might be things like recessive genetic abnormalities, right? Picture a really fantastic bull with excellent genetic merit in a lot of different traits, and some people might be a little reluctant to use that particular individual because they're a carrier for a recessive genetic abnormality, right? We could actually go in and correct that genetic abnormality and have the exact same animal without the abnormality, right? So lots of potential there. We also might be able to target some traits that have mutations that we know of that have large effects. So things like tenderness with calpain and calpastatin, or growth hormone or its receptor. Potentially something like DGAT, which affects milk fat and milk yield in dairy cattle. The SLIC mutation, which is a mutation in prolactin receptor, I believe, that makes cattle shed their hair off. They don't get the really big woolly hairy coat. So all of these things are sort of large effect mutations that would be really good targets for this type of technology. Probably not going to be very effective for things that are really, really small effect because we'd have to make too many edits to make it cost effective at this point in time, and B, a lot of those we don't actually know the causal mutation. So you have to know the mutation that causes the desired phenotype to be able to utilize this technology. So here's where I think this kind of gets interesting, and that is when we start thinking about an incorporation of this technology and how we can combine it with other types of technologies to really accelerate genetic progress. So in this particular example, I'm not going to go through every single thing on here, but it sort of starts with a particular mating that produces a bunch of embryos. They transfer the embryos and collect the fetuses, and you basically create a bunch of different cell lines. From there, you can use the genotyping technologies that we have to try and identify the cell line that has the best genetic merit. And then we'd go through a process, and we might take the best cell lines and do some editing within that cell line. Maybe change some alleles in those individuals for some of those large effect traits, or correct a line that may be a carrier of recessive abnormality. Those are all types of things we could do with that technology. And then you go in and use those things in somatic cell nuclear transfer, and end up with these high genetic merit animals that have been genome edited within a fairly short amount of time. And when I sort of think about this, I stole the original slide from Mark Allen at Transova, but I think when we start to think about the pairing of genome editing technologies with our advanced reproductive technologies, I think it really becomes interesting and sort of fascinating with what we might be able to accomplish with these types of technologies. Because one of the challenges with beef cattle production and genetic selection is they have long generation intervals, right? We can do phenomenal things with the tools that we have, but it's going to take us some time to do that. And that's a challenge that pork producers and poultry producers don't necessarily share, right? They can do turnover generations much more quickly than we can. But if we start to think about genomically selecting superior animals, then maybe we do some IVF, and we create hundreds of different embryos. We could use some of those in embryo transfer. Others we could establish basically cell lines out of those embryos. We could genotype them, identify the superior ones, and edit some of those and maybe use some of those in a cloning application. They could also be used to produce gametes that could be used in AI or IVF, and then sort of continue on around this circle. So we might be able to go around this circle a couple of different times pretty rapidly within a few months and go through a couple of generations really, really quickly with this type of technology. So using our reproductive technologies as the base, and then adding the genome editing technologies to it. So the two main challenges that I see for this type of technology is, of course, consumer acceptance, right? We've all seen that sometimes there is a little bit of pushback on something that might be considered genetically modified, right? So to sort of combat this, I actually think they've gone about it in a way that is extremely clever. So if you notice some of those proof of concepts, namely Spotty Guy and Burry, right? They sort of tackled a trait first that should have widespread consumer acceptance, right? If we're not dehorning those animals, if we're not dehorning millions of dairy cattle, right? That's probably a lot fewer painful procedures for those animals. That's something consumers can probably get behind, right? And in fact, if you look at some of the articles discussing this, you'll actually see that they have people from the Humane Society of the United States that have signed on in support of this technology because of the type of trait that they sort of tackled first. The second challenge is, of course, FDA approval guidelines. So you may or may not know that when we do genetic modifications in animals, that is governed by the FDA. And at this point in time, any modifications to the genome using those technologies would be considered a drug. So it would have to go through the same sort of approval process as like your cholesterol medication or any other type of drug that we might give someone, right, to prove that it's safe. And that's very different than what the plant folks have to do, but that's what we have to do in animals. So as of late last year, they put out some draft guidelines specifically to address sort of this genome editing technology, and there weren't really any changes. That was still going to be regulated as a drug. As I understand it, there was a fairly large pushback from the scientific community, and they're still reviewing those guides at this point in time. The last draft I've seen was April of this year, and they still hadn't changed anything, but there are some committees, and they've been talking about ways that they might address this. So if some of the rules governing its use could change so that it wouldn't be so cost prohibitive to get these types of things approved, we might be able to use a technology like this as the little cherry on top of our sundaes in the beef industry. And with that, I can turn it back over to AJ.
And unfortunately, Dr. Rolf has to run out of here before we have a chance Q&A at the end of today. So if anybody has any questions, be happy to try to get those fielded right now.